In this video, we'll cover everything within the homeostasis and response topic, from how your body maintains stable internal conditions to the nervous and hormonal systems that control your responses, and how plants use hormones to grow and respond to their surroundings. In this lesson, we'll begin by examining what homeostasis is and why it's essential for our survival. Then look at how automatic control systems in the body maintain this stability, explore the concept of negative feedback as the key mechanism behind homeostasis, and finally introduce some examples of homeostatic systems in the human body. Homeostasis comes from Greek words meaning staying the same. It refers to the process of maintaining a stable internal environment within our bodies. The conditions inside your body need to be kept steady, even when the external environment changes. This stability is crucial for survival. Your cells need the right conditions to function properly. This includes the right conditions for enzyme activity, which we've discussed in previous episodes. Homeostasis is all about the regulation of the conditions inside your body. It involves maintaining a stable internal environment in response to changes in both internal and external conditions. Our bodies have several automatic control systems that regulate our internal environment. These include both nervous and hormonal communication systems. For example, there are control systems that maintain your body temperature, blood glucose level, and water content. All your automatic control systems are made up of three main components that work together to maintain a steady condition. The first component is the receptor. Receptors detect stimuli. They monitor levels of substances or conditions in your body. The second component is the coordination center. This receives and processes the information from the receptors, then organizes a response. The third component is the effector. Effectors produce a response that counteracts any change and helps restore the optimum level. Your automatic control systems keep your internal environment stable using a mechanism called negative feedback. When the level of something like water or temperature gets too high or too low, your body uses negative feedback to bring it back to normal. Let's look at how this works when levels are too high. First, the receptor detects the stimulus, that the level is too high. Next, the coordination center receives and processes this information, then organizes a response. Then the effector produces a response which counteracts the change and restores the optimum level. The level decreases. It's important to understand that the effectors will continue producing responses as long as they're stimulated by the coordination center. This might cause the level to become too much in the opposite direction. Luckily, the receptor detects if the level becomes too different and triggers the negative feedback loop again. This process happens without you thinking about it. It's completely automatic. Your body is constantly making these adjustments to maintain homeostasis. Examples of homeostatic systems in the human body include regulation of body temperature, blood glucose levels, and water content. We'll explore each of these systems in detail in upcoming episodes, looking at the specific receptors, coordination centers, and effectors involved. In our next episode, we'll take a closer look at the nervous system, one of the key systems involved in homeostatic control. In this episode, we'll start by examining the basic function of the nervous system and why it's so important, then look at the different parts that make it up, explore how these components work together to coordinate responses, and finally investigate reflex actions and how they help protect us from harm. All organisms need to respond to stimuli, changes in their environment, in order to survive. Single-celled organisms can simply respond directly to their environment, but in multicellular organisms like humans, Cells need to communicate with each other first. As organisms evolved and became more complex, they developed nervous and hormonal communication systems. The nervous system is what allows humans to react to their surroundings and coordinate their behavior. It detects stimuli, processes information, and coordinates responses. Let's look at the different parts that make up this complex system. The nervous system can be divided into two main parts. First is the central nervous system, or CNS, which consists of the brain and spinal cord. In vertebrates, animals with backbones, the CNS is protected by bone, the brain is enclosed in the skull, and the spinal cord runs through the vertebral column. The second part is the peripheral nervous system. This consists of sensory neurons and motor neurons that connect the CNS to the rest of the body. Neurons are specialized cells that carry electrical impulses. There are different types of neurons with different functions. Sensory neurons carry information as electrical impulses from receptors to the CNS. Motor neurons carry electrical impulses from the CNS to effectors, the parts of the body that respond to commands from the nervous system. Receptors are cells that detect stimuli. There are many different types of receptors specialized for different stimuli. For example, we have taste receptors on the tongue and sound receptors in the ears. Receptors can form part of larger, complex organs. The retina of the eye is covered in light receptor cells that detect light of different wavelengths. Effectors respond to nervous impulses and bring about a change. Muscles and glands are known as effectors. They respond in different ways. Muscles contract in response to a nervous impulse, whereas glands secrete hormones. The central nervous system acts as a coordination center. It receives information from the receptors and then coordinates a response, which is carried out by effectors. This whole process happens very quickly thanks to the way neurons communicate with each other. Neurons transmit information very quickly to and from the brain, and your brain quickly decides how to respond to a stimulus. The connection between two neurons is called a synapse. Here, the nerve signal is transferred by chemicals which diffuse across the gap. These chemicals then set off a new electrical signal in the next neuron. This allows information to pass from one neuron to the next throughout the nervous system. But some responses happen even more quickly through reflexes. Reflexes are rapid, automatic responses to certain stimuli that don't involve the conscious part of the brain. Reflexes help reduce the chances of being injured. For example, if someone shines a bright light in your eyes, your pupils automatically get smaller so that less light gets into the eye. The passage of information in a reflex from receptor to effector is called a reflex arc. When a stimulus, such as a painful bee sting, is detected by receptors, impulses are sent along a sensory neuron to a relay neuron in the CNS. When the impulses reach a synapse between the sensory neuron and the relay neuron, they trigger chemicals to be released. These chemicals cause impulses to be sent along the relay neuron. When the impulses reach a synapse between the relay neuron and a motor neuron, the same thing happens. Chemicals are released and cause impulses to be sent along the motor neuron. The impulses then travel along the motor neuron to the effector, which is usually a muscle in this example. The muscle then contracts and moves your hand away from the bee. Because you don't have to think about this response, it happens very quickly, helping to minimize damage from harmful stimuli. Understanding the nervous system helps us appreciate how our bodies detect and respond to the world around us. This complex network of cells allows us to react quickly to changes, protecting us from harm and helping us interact with our environment. In our next episode, we'll explore specific parts of the nervous system in more detail, looking at how the brain processes information and coordinates complex responses. Let's begin by going over exactly what we'll cover in this video. First, we'll look at the brain's role as part of the central nervous system. Next, we'll examine different regions of the brain and their specific functions. Then we'll explore the methods scientists use to study the brain. Finally, we'll discuss some of the challenges in investigating and treating brain disorders. Along with the spinal cord, the brain forms part of the central nervous system. It's made up of billions of interconnected neurons, specialized cells that communicate through electrical and chemical signals. The brain is in charge of all our complex behaviors. It controls and coordinates everything we do. Different regions of the brain carry out different functions. Understanding these regions helps us appreciate how this complex organ works. The cerebral cortex is the outer wrinkly layer of the brain. This region is responsible for higher functions like consciousness, intelligence, and language. The cerebellum is located at the back of the brain and is primarily responsible for muscle coordination. It helps ensure our movements are smooth and precise. The medulla is a region that controls unconscious activities, things you don't have to think about doing, like breathing and regulating your heartbeat. Scientists use a range of methods to study the brain and map out which bits do what. One approach involves studying patients with brain damage. If a small part of the brain has been damaged, the effect this has on the patient can tell us a lot about what the damaged part of the brain does. For example, if an area at the back of the brain was damaged by a stroke and the patient went blind, you know that this area has something to do with vision. The brain can also be studied by electrical stimulation. By pushing a tiny electrode into the tissue and giving it a small zap of electricity, scientists can observe what happens. When different parts of the brain tissue are stimulated, it's possible to get an idea of what those parts do. For instance, when a certain part of the brain, known as the motor area, is stimulated, it causes muscle contraction and movement. Magnetic resonance imaging, MRI scanners, are advanced machines that can produce very detailed pictures of the brain structures. Scientists use these to find out what areas of the brain are active when people are doing things like listening to music or trying to remember something. Knowledge of how the brain works has led to the development of treatments for disorders of the nervous system. For example, electrical stimulation of the brain can help reduce muscle tremors caused by nervous system disorders, such as Parkinson's disease. However, the brain is incredibly complex and delicate. This makes the investigation of brain function and the treatment of brain damage or disease challenging. It also carries risks, such as physical damage to the brain during surgery. This means treatments must be developed and applied with extreme caution. Neuroscientists, scientists who study the brain, continue to develop new technologies and methods to better understand this complex organ. Each advancement brings us closer to understanding how the brain works and how to treat neurological disorders more effectively. In our next episode, we'll explore another fascinating sensory organ, the eye, and discover how it allows us to perceive the visual world around us. Let's begin by going over exactly what we'll cover in this video. First, we'll examine the structure of the eye and the function of its various parts. Next, we'll look at how the iris reflex works to protect our eyes in different lighting conditions. Then we'll explore the process of accommodation, how our eyes focus on both near and distant objects. Finally, we'll discuss common vision defects and the ways they can be corrected. The eye is a sophisticated sense organ with several important components working together to enable vision. Let's look at these key structures. The sclera forms the tough, white exterior wall of the eye. This protective layer helps maintain the eye's shape and provides structural support. At the front of the eye sits the cornea. a clear dome-shaped window that begins the process of bending light as it enters the eye. Behind the cornea is the iris, the colorful circular structure that contains muscles. These muscles control the size of the pupil, which is the opening in the center of the iris that allows light to pass through. The lens sits behind the pupil and further directs light toward the back of the eye. It focuses incoming light rays onto the retina, which contains cells that respond to light intensity and different colors. The ciliary muscles and suspensory ligaments work together to adjust the lens shape, allowing us to see objects at varying distances clearly. The optic nerve connects the eye to the brain, transmitting visual information from the light-sensitive cells in the retina to the visual processing centers in the brain. Our eyes have developed sophisticated mechanisms to protect themselves from damage. One important example is the iris reflex, which adjusts our eyes for different light conditions. Intense light can harm the delicate retina, so a protective reflex exists. When excessive brightness is detected by light receptors, a reflex pathway activates that reduces pupil size. This occurs because circular muscles within the iris tighten while the radial muscles loosen. This adjustment limits the amount of light entering the eye, safeguarding the retina from potential damage. In dim environments, the opposite reaction takes place. The radial muscles tighten while the circular muscles relax, causing the pupil to expand. This automatic adjustment optimizes our vision in varying light conditions, allowing more light in when it's dark and restricting light when it's bright. Another crucial function of the eye is focusing on objects at different distances, a process called accommodation. The eye achieves proper focus by altering the lens shape. When viewing nearby objects, a sequence of changes occurs. The eye's ciliary muscles tighten, causing the tension on the suspensory ligaments to decrease. With reduced tension, the naturally elastic lens becomes more curved or rounded. This increased curvature bends light rays more sharply, bringing close objects into focus on the retina. For distant vision, the process reverses. The ciliary muscles loosen, allowing the suspensory ligaments to become taut. This tension pulls on the lens, making it flatter and less curved. The flattened lens bends light less dramatically, allowing distant objects to focus properly on the retina. These adjustments ensure clear vision whether we're reading a book or looking at the horizon. Vision problems develop when the eye cannot properly focus light on the retina. The two most common vision conditions are hyperopia and myopia. People with hyperopia, long-sightedness, can see distant objects clearly but struggle with close-up vision. This happens when the eyeball is shorter than normal or when the lens doesn't curve enough. In these cases, the focal point falls behind the retina rather than directly on it. This creates blurry vision when looking at nearby objects like books or screens. The clinical term for this condition is hyperopia. It can be corrected with convex lenses that curve outward in the middle. These lenses help bend light rays more sharply before they enter the eye, shifting the focal point forward onto the retina. People with myopia, short-sightedness, experience the reverse problem. They see nearby objects clearly but have difficulty with distant vision. This occurs when the eyeball is too elongated or when the lens curves too much. This causes the focal point to fall in front of the retina rather than on it, making distant objects appear blurry. The clinical term for this condition is myopia. It can be addressed using concave lenses that curve inward in the middle. These lenses help spread light rays before they enter the eye, moving the focal point backward onto the retina. Several treatments exist for vision correction beyond traditional eyeglasses. Understanding these options provides important context for those with vision impairments. Contact lenses are small optical devices that rest directly on the eye's surface. They're designed to address specific vision problems by altering how light enters the eye. Many individuals prefer contacts because they're virtually invisible and don't interfere with physical activities. They provide a more natural field of vision compared to glasses. They come in two main varieties: rigid gas permeable lenses and soft lenses. While soft versions offer greater comfort, they present a somewhat higher infection risk than their rigid counterparts. Laser eye correction has become increasingly common for treating vision problems. This surgical approach uses precise laser technology to reshape the corneal tissue. By carefully modifying the cornea's curvature, surgeons can adjust how the eye bends incoming light. This technique can effectively address nearsightedness, farsightedness, and astigmatism. The procedure allows for remarkable precision, often eliminating the need for corrective lenses completely. However, as with any surgical intervention, there are potential risks, including dry eyes, infection, or occasional visual disturbances. For certain patients, particularly those with advanced hyperopia, lens replacement surgery might offer better results than laser treatment. This procedure involves removing the natural lens and replacing it with a custom designed artificial implant made of biocompatible materials. This approach resembles cataract surgery but focuses on vision correction rather than removing clouded lenses. The synthetic lens can be specifically designed to address the patient's unique vision requirements. Understanding how the eye works helps us appreciate the complexity of this remarkable organ. From automatically adjusting to different light levels to focusing on objects at varying distances, the eye performs incredibly sophisticated functions with no conscious effort from us. The ability to correct vision defects has transformed millions of lives, allowing people to see clearly regardless of the natural capabilities of their eyes. Let's begin by going over exactly what we'll cover in this video. First, we'll examine why maintaining a constant body temperature is so important. Next, we'll look at the thermoregulatory center in the brain and how it monitors temperature. Then we'll explore the body's responses to being too hot. Finally, we'll discover the mechanisms that help us warm up when we're too cold. The human body needs to keep its internal temperature at around 37 degrees Celsius. This is the optimum temperature for enzymes in the body to function properly. If body temperature rises or falls too much, enzymes may stop working effectively or even become permanently damaged. This can disrupt vital chemical reactions and potentially be life-threatening. To maintain this precise temperature, the body must balance the energy gained through respiration with the energy lost to the environment. This balance is controlled by the thermoregulatory center in the brain. Located in a region called the hypothalamus, this center contains receptors that are sensitive to the temperature of blood flowing through the brain. The thermoregulatory center also receives information from temperature receptors in the skin. These provide early warning of changes in external temperature before they affect the core body temperature. This system works as a classic example of a negative feedback mechanism. When body temperature deviates from the normal range, responses are triggered that counteract the change. When temperature receptors detect that core body temperature is too high, the thermoregulatory center acts as a coordination center. It processes this information and automatically triggers appropriate effectors. These effectors, such as sweat glands, produce responses that counteract the change in temperature. Similarly, when temperature receptors detect that core body temperature is too low, the thermoregulatory center triggers different effectors, such as muscles, to produce responses that warm the body. Some effectors work antagonistically. One effector heats and another cools, working simultaneously to achieve a very precise temperature. This mechanism allows for a more sensitive response than a simple on-off system. Let's look more closely at the specific responses produced by effectors to counteract changes in body temperature. When you're too hot, several mechanisms activate to help cool you down. First, sweat is produced by sweat glands and evaporates from the skin. This evaporation process transfers energy to the environment, cooling the body. Second, the blood vessels supplying the skin dilate, becoming wider. This is called vasodilation. Vasodilation allows more blood to flow close to the surface of the skin. This helps transfer heat energy from deeper tissues to the skin where it can be lost to the surroundings. These cooling mechanisms are particularly effective when the air is dry, promoting evaporation, and when there is a temperature difference between the body and the environment. When you're too cold, a different set of responses occur to conserve and generate heat. First, the hairs on your skin stand up. This is often called goosebumps and creates a layer of trapped air that acts as insulation. Second, sweat production stops. This prevents heat loss through evaporation. Third, blood vessels supplying skin capillaries constrict, becoming narrower. This is called vasoconstriction. Vasoconstriction reduces blood flow to the skin surface. This minimizes heat loss and helps maintain the temperature of vital internal organs. Fourth, when you're very cold, your muscles contract rapidly and involuntarily in a process called shivering. This increased muscle activity generates heat through respiration, helping to warm the body. These heating and cooling mechanisms work together to maintain our core temperature within a narrow range, despite varying external conditions. Both sets of responses are automatic. We don't consciously control them. They're controlled by the autonomic nervous system, which regulates many unconscious bodily functions. Understanding temperature regulation helps us appreciate the sophisticated control systems in our bodies. This homeostatic mechanism demonstrates how different parts of the body, the brain, skin, blood vessels, and muscles work together to maintain the stable internal conditions necessary for life. Let's begin by going over what we'll cover. First, we'll examine what hormones are and how they function. Next, we'll look at the major endocrine glands and their roles. Finally, we'll compare hormonal and nervous communication. Our bodies need to send information between different parts to coordinate activities. While the nervous system uses electrical impulses traveling along neurons, the endocrine system uses chemical messengers called hormones. Hormones are chemical molecules released directly into the bloodstream. They travel throughout the body but only affect particular cells in specific organs, called target organs. These target cells have special receptors that recognize and respond to specific hormones. Unlike the rapid but short lived signals of the nervous system, hormones typically create slower but longer lasting effects. Hormones are produced by specialized structures called endocrine glands. These glands secrete their hormones directly into the bloodstream rather than through ducts. The pituitary gland, located at the base of the brain, is often called the master gland because it produces many hormones that regulate body conditions and control other glands. It directs them to release hormones that bring about changes throughout the body. The thyroid gland in the neck produces thyroxine, which regulates metabolism, heart rate, and temperature. This hormone controls how quickly the body uses energy. The adrenal glands, situated on top of the kidneys, produce adrenaline. This hormone prepares the body for fight or flight responses during stress or danger. The pancreas produces insulin, which regulates blood glucose levels. We'll explore this crucial hormone in more detail in our next episode. In females, the ovaries produce estrogen, which is involved in the menstrual cycle and secondary sexual characteristics. In males, the testes produce testosterone, which controls puberty and sperm production. While both the nervous and hormonal systems help coordinate body functions, they work quite differently. The nervous system acts very quickly, but for a short time, affecting precise areas of the body. For example, if you touch something hot, nerve signals rapidly travel to and from your brain, causing you to pull away immediately. The endocrine system acts more slowly but has longer lasting effects, influencing the body in a more general way. When you experience stress, adrenaline is released gradually but continues affecting your body for some time. If you're not sure whether a response is nervous or hormonal, consider its timing and duration. Very quick responses are typically nervous, while those lasting longer are usually hormonal. Understanding the endocrine system helps us appreciate how our bodies maintain balance through chemical signals. These hormones ensure our cells work together harmoniously despite changing internal and external conditions. In our next episode, we'll explore one of the most important hormonal systems, insulin and the control of blood glucose. Let's begin by going over what we'll cover. First, we'll examine what affects blood glucose levels in our bodies. Next, we'll look at how insulin and glucagon work together to maintain balance. Finally, we'll explore what happens when this control system doesn't work properly in diabetes. Our blood glucose concentration needs careful regulation. Too much or too little glucose in the bloodstream can cause serious health problems. The pancreas is the key organ responsible for monitoring and controlling blood glucose levels. It produces two important hormones, insulin and glucagon, that work in opposition to keep blood glucose levels stable. Several factors affect the amount of glucose in our blood. When we eat foods containing carbohydrates, these are digested, and glucose enters our bloodstream from the intestines. At the same time, normal cell metabolism constantly removes glucose from the blood as cells use it for energy. Physical activity, especially vigorous exercise, increases the rate at which glucose is removed from the blood, as active muscles require more energy. When we have excess glucose, it can be stored as glycogen in the liver and muscles for later use. The liver can store a significant amount of glycogen, making it a crucial organ for blood glucose regulation. The level of glucose in the blood must be kept steady. The normal range is quite narrow, and deviations can have serious consequences. This is why the body has developed sophisticated mechanisms to monitor and control blood glucose levels. The body uses a negative feedback system to maintain blood glucose within the normal range. This means that any change away from the normal range triggers responses that counteract the change, bringing levels back to normal. When blood glucose levels rise too high, perhaps after eating a meal rich in carbohydrates, the pancreas detects this change and responds by secreting insulin into the bloodstream. Insulin causes liver cells to convert glucose into glycogen for storage. This removes excess glucose from the blood, bringing levels back down to normal. Insulin also helps muscle and fat cells absorb more glucose from the bloodstream, further reducing blood glucose levels. It does this by causing special glucose transporters to move to the cell membrane, allowing glucose to enter the cells. The overall effect of insulin is to reduce blood glucose concentration by moving glucose from the blood into cells, where it can be used or stored. Conversely, when blood glucose levels fall too low, perhaps during exercise or when we haven't eaten for a while, the pancreas detects this change and secretes glucagon. Glucagon has essentially the opposite effect to insulin. It causes liver cells to convert stored glycogen back into glucose, which is released into the bloodstream. This raises blood glucose levels back to normal. These two hormones, insulin and glucagon, work together in a balanced way to maintain steady blood glucose levels, regardless of whether we've just eaten or haven't had food for hours. This is a classic example of antagonistic hormone action in maintaining homeostasis. Unfortunately, this control system can fail, resulting in a condition called diabetes. There are two main types of diabetes, each with different causes but similar effects. In type 1 diabetes, the pancreas cannot produce enough insulin. This is usually caused by an autoimmune reaction where the body's immune system attacks and destroys the insulin producing cells in the pancreas. Without insulin, glucose cannot enter the body's cells and remains in the bloodstream, leading to high blood glucose levels. People with type 1 diabetes need regular insulin injections throughout their lives to control their blood glucose levels. The amount of insulin required depends on several factors, including diet, physical activity levels, and individual metabolism. Modern insulin therapy can be very effective, allowing people with type 1 diabetes to live normal, healthy lives. Type 2 diabetes occurs when body cells become resistant to insulin. The pancreas still produces insulin, sometimes even in larger than normal amounts, but cells don't respond properly to it. This means glucose stays in the blood rather than being taken up by cells, causing high blood glucose levels. Being overweight increases the risk of developing type 2 diabetes, as does lack of regular exercise. Type 2 diabetes often develops gradually, and many people have the condition for years before it's diagnosed. Early symptoms can include increased thirst, frequent urination, fatigue, and blurred vision. Treatment for type 2 diabetes usually involves lifestyle changes, such as improved diet and increased exercise. In more severe cases, medication to increase insulin sensitivity or stimulate insulin production may be needed. Understanding glucose regulation helps us appreciate the complex balancing act our bodies perform constantly. This homeostatic mechanism is critical for maintaining the stable internal environment our cells need to function properly. In our next episode, we'll explore other hormones and their roles in regulating different aspects of body function. Let's begin by going over exactly what we'll cover in this video. First, we'll examine the basic structure and function of the kidneys. Next, we'll look at how the kidneys filter blood and remove waste products. Then we'll explore how water balance is maintained through hormonal control. Finally, we'll investigate what happens when kidneys fail and the treatments available. The kidneys are bean-shaped organs located towards the back of the abdominal cavity, one on each side of the spine. Though relatively small, they perform several vital functions that maintain homeostasis in the body. The primary role of the kidneys is to filter waste products and excess substances from the blood. This cleaning process is essential for removing potentially harmful compounds while retaining useful materials. When blood passes through the kidneys, a complex filtration process occurs. This process is remarkably selective, allowing the kidneys to remove specific substances while returning others to the bloodstream. This filtration happens in microscopic structures called nephrons, which are the functional units of the kidneys. Each kidney contains about 1 million nephrons, giving them tremendous filtering capacity. The first stage of kidney function is ultrafiltration, where small molecules are filtered out of the blood as it passes through tiny blood vessels in the kidneys. The second stage is selective reabsorption, where useful substances are reabsorbed back into the bloodstream. This ensures that valuable materials aren't wasted by being excreted. The substances that aren't reabsorbed are removed from the body in urine, which is stored in the bladder before being expelled. Let's look more closely at the specific waste products removed by the kidneys. The first major waste product is urea. Urea forms when proteins are broken down in the liver. Proteins contain nitrogen, and when excess amino acids are broken down, this nitrogen needs to be removed from the body. Proteins and amino acids can't be stored by the body, unlike fats and carbohydrates. When we consume more protein than needed for growth and repair, The excess amino acids are converted to urea in a process called deamination. Ammonia is actually produced as an initial waste product during this process, and it's highly toxic. The liver quickly converts ammonia to urea, which is much less harmful and can be safely transported in the bloodstream to the kidneys for excretion. The kidneys also regulate ion levels in the blood. Ions such as sodium are taken into the body in food and then absorbed into the bloodstream. If the ion content of the body is wrong, this could upset the balance between ions and water, making it difficult to draw water into cells by osmosis. Having the wrong amount of water can damage cells or mean they don't work normally. Some ions are lost in sweat, which tastes salty due to its sodium content. However, the amount of ions in the body is primarily regulated by the kidneys. The right amount of ions must be maintained in the blood after filtration, and the rest is removed from the body in urine. Perhaps the most critical function of the kidneys is regulating water balance. Our bodies are approximately 70% water, and maintaining the correct amount is essential for all cellular functions. The body has to constantly balance the water coming in against the water going out. We lose water from the skin through sweat and from the lungs when breathing out. We can't control how much water we lose in these ways, so the amount of water is balanced by the amount we consume and the amount removed by the kidneys in urine. A specific hormone known as antidiuretic hormone regulates how concentrated our urine becomes. The pituitary gland, located at the base of the brain, secretes this hormone into the circulatory system. Special receptors in the brain continuously measure blood water levels, sending signals to the pituitary to adjust ADH secretion as needed. The whole process of water content regulation is controlled by negative feedback. This means that if the water content gets too high or too low, a mechanism will be triggered that brings it back to normal. When water content in the blood is too low, receptors in the brain detect this change. The coordination center in the brain receives this information and coordinates a response. The pituitary gland releases more ADH, so more water is reabsorbed from the kidney tubules back into the blood. This results in a smaller volume of more concentrated urine. Conversely, when water content in the blood is too high, the brain receptors detect this change. The coordination center receives this information and coordinates a different response. The pituitary gland releases less ADH, so less water is reabsorbed from the kidney tubules. This results in a larger volume of more dilute urine, removing the excess water from the body. This precise control mechanism allows our bodies to adapt to varying water intake and environmental conditions while maintaining a stable internal environment. What happens if the kidneys stop working properly? If someone's kidneys fail, waste products build up in their blood, and they lose the ability to control the levels of ions and water in their body. Eventually, this leads to death if not treated. There are two main treatments for kidney failure: regular dialysis or a kidney transplant. Dialysis is a medical procedure that artificially filters the blood. It's designed to keep concentrations of dissolved substances in the blood at normal levels and to remove waste products. In a dialysis machine, the patient's blood flows between partially permeable membranes surrounded by dialysis fluid. This fluid has the same concentration of dissolved ions and glucose as healthy blood. This means these substances won't be lost from the blood during dialysis. However, waste products, such as urea, are absent from the dialysis fluid. This creates a concentration gradient that causes urea and excess ions and water to diffuse across the membrane from the blood into the dialysis fluid. Many patients with kidney failure have to undergo dialysis sessions 3 times a week, with each session taking 3 to 4 hours. It's not a pleasant experience and is expensive to provide, but dialysis can buy patients with kidney failure valuable time until a donor organ is found. The alternative treatment is a kidney transplant, which is currently the only cure for kidney failure. Healthy kidneys for transplant typically come from people who have died suddenly and were on the organ donor register or carried a donor card. Kidneys can also be donated by living people, as we only need one functioning kidney to survive. However, there is a small risk to the person donating the kidney. One challenge with transplants is that the donated kidney can be rejected by the patient's immune system. To prevent this, patients must take immunosuppressant drugs, which reduce the activity of their immune system. While these drugs help prevent rejection, they also make the patient more vulnerable to infections. Despite these challenges, transplants are generally cheaper in the long run than dialysis and put an end to the hours patients would otherwise spend on dialysis machines. Understanding how the kidneys maintain water and nitrogen balance helps us appreciate the complex systems that keep our bodies functioning properly. These remarkable organs work continuously behind the scenes, filtering our blood about 60 times each day and producing about 1 to 2 liters of urine. In our next episode, we'll explore the role of hormones in human reproduction and how they control the development of secondary sexual characteristics and the menstrual cycle. To get started, let me walk you through what we'll be covering. First, we'll examine how hormones trigger the development of secondary sexual characteristics during puberty. Next, we'll look at the main reproductive hormones in males and females. Then we'll explore the menstrual cycle in detail, including its four main stages. Finally, we'll investigate how different hormones control this cycle. At puberty, the body undergoes significant changes as it develops from a child into an adult capable of reproduction. These changes are triggered by sex hormones. In males, the main reproductive hormone is testosterone. This hormone is produced by the testes and stimulates sperm production. Testosterone also triggers the development of secondary sexual characteristics, such as facial hair. In females, the main reproductive hormone is estrogen. It's produced by the ovaries and brings about physical changes, such as breast development. Estrogen is also involved in the menstrual cycle and helps eggs mature. These hormones don't just appear suddenly at puberty. They're actually released in very small amounts throughout childhood, but their levels increase dramatically during the teenage years. The menstrual cycle is a complex series of changes that occurs in a woman's reproductive system. Each cycle takes approximately 28 days, though this can vary between individuals. The cycle involves the release of an egg from a woman's ovaries and the preparation of the uterus to receive a fertilized egg. If fertilization doesn't occur, the uterus lining breaks down and is shed. The menstrual cycle can be divided into four main stages. Stage 1 is menstruation, where the uterus lining breaks down for about 4 days. Stage 2 involves the rebuilding of the uterus lining. From day 4 to day 14, a thick spongy layer full of blood vessels forms, ready to receive a fertilized egg. Stage 3 occurs around day 14, when an egg develops and is released from the ovary. This is called ovulation. In stage 4, the uterus wall is then maintained for about 14 days until day 28. If no fertilized egg has implanted in the uterus wall by this time, the spongy lining starts to break down and the cycle begins again. This cycle is controlled by a complex interplay of four hormones: follicle stimulating hormone, luteinizing hormone, estrogen, and progesterone. Follicle stimulating hormone is produced in the pituitary gland. It causes an egg to mature in one of the ovaries, specifically in a structure called a follicle. Follicle stimulating hormone also stimulates the ovaries to produce estrogen, which in turn causes the lining of the uterus to grow. Estrogen has additional roles in the cycle. It stimulates the release of luteinizing hormone, and inhibits the release of follicle stimulating hormone. Luteinizing hormone is also produced by the pituitary gland. It stimulates the release of an egg at day 14 of the cycle, the process known as ovulation. After ovulation, the remains of the follicle develop into a structure called the corpus luteum, which produces progesterone. Progesterone maintains the lining of the uterus during the second half of the cycle. If progesterone levels fall, the lining breaks down, leading to menstruation. Progesterone also inhibits the release of luteinizing hormone and follicle stimulating hormone, preventing further eggs from developing during the current cycle. This entire system works through negative feedback loops. For example, when estrogen levels rise, they inhibit follicle stimulating hormone production, preventing additional eggs from maturing. When progesterone levels rise, they inhibit luteinizing hormone, preventing additional ovulations. If a fertilized egg implants in the uterus wall, it produces hormones that maintain the lining, preventing its breakdown and the start of a new cycle. This is one of the first signs of pregnancy. Understanding these hormonal processes is important not just for appreciating the complexity of human reproduction, but also for developing contraceptive methods and treatments for various reproductive disorders. The precise timing and interaction of these hormones ensure that the reproductive system functions properly, preparing the body each month for the possibility of pregnancy. In our next episode, we'll explore how an understanding of these hormonal mechanisms has led to the development of various contraceptive methods that help people plan their families. Let's start by outlining the main points we'll go over. First, we'll briefly review how pregnancy occurs. Next, we'll examine how hormonal methods of contraception work. Then, we'll look at various barrier methods that prevent sperm and egg meeting. Finally, we'll explore some other approaches to preventing pregnancy. Pregnancy happens when a sperm cell reaches and fertilizes an egg that has been released from the ovary during ovulation. Contraception methods aim to prevent this process in different ways. Hormones can be used as an effective method of contraception. These work by disrupting the normal reproductive cycle we discussed in our previous episode. Estrogen can be used to prevent egg release from the ovaries. This might seem counterintuitive since estrogen naturally helps stimulate egg release. However, when taken continuously in contraceptive form, estrogen actually prevents the production of follicle stimulating hormone. Without follicle stimulating hormone, egg development and maturation stop and ovulation doesn't occur. Progesterone also reduces fertility in several ways. It stimulates the production of thick cervical mucus, which creates a barrier that makes it difficult for sperm to swim through and reach an egg. Additionally, progesterone thins the uterus lining, making it less suitable for a fertilized egg to implant. Many contraceptive methods use combinations of these hormones. The combined oral contraceptive pill contains both estrogen and progesterone. This pill is over 99% effective at preventing pregnancy when used correctly. However, it can cause side effects like headaches and mood changes, and doesn't protect against sexually transmitted diseases. The progesterone only pill has fewer side effects than the combined pill and is just as effective when taken properly. Other hormonal contraceptive methods include the contraceptive patch, which contains estrogen and progesterone. This small patch sticks to the skin, and each one lasts for 1 week. The contraceptive implant is inserted under the skin of the arm. It releases a continuous amount of progesterone over an extended period, stopping egg release and thickening cervical mucus. Contraceptive injections also contain progesterone, and each dose lasts 2 to 3 months. Intrauterine devices are T-shaped devices inserted into the uterus. Some types release hormones like progesterone, while copper intrauterine devices don't contain hormones but still prevent pregnancy by creating an environment hostile to sperm. Non-hormonal contraception methods work by creating physical barriers that stop sperm from reaching the egg. Condoms are worn over the penis during intercourse to prevent sperm entering the vagina. There are also female condoms that line the inside of the vagina. Both types of condoms are the only form of contraception that offer protection against sexually transmitted diseases. A diaphragm is a shallow plastic cup that fits over the cervix, forming a barrier. It must be used with spermicide, a substance that disables or kills sperm. Spermicide can also be used alone as a form of contraception, but it's not as reliable as when combined with other methods. There are other approaches to avoiding pregnancy beyond hormonal and barrier methods. Sterilization involves cutting or tying the fallopian tubes in females or the sperm duct in males. This is a permanent procedure with a very small chance that the tubes can rejoin. Some people use natural methods to avoid pregnancy. This involves tracking when the woman is most fertile during her menstrual cycle and avoiding intercourse on those days. While popular with people who prefer not to use hormonal or barrier methods, natural timing is less effective than most other contraception methods. Abstinence, not having sexual intercourse, is the only method that is 100% effective at preventing pregnancy. Each contraceptive method has advantages and disadvantages regarding effectiveness, ease of use, potential side effects, and protection against sexually transmitted diseases. Understanding how different contraceptive methods work allows people to make informed choices about their reproductive health and family planning. In our next episode, we'll explore how hormones can be used to treat infertility when people have children but are experiencing difficulties conceiving naturally. Before we get into the details, here's what's coming up. First, we'll examine how hormones can be used to increase fertility. Next, we'll look at in vitro fertilization and how it works. Then we'll explore the benefits and challenges of fertility treatments. Finally, we'll briefly consider some of the ethical questions surrounding these technologies. Some women experience fertility problems because their hormone levels aren't optimal for conception. For example, they may have levels of follicle stimulating hormone that are too low to trigger egg maturation. When eggs aren't properly released from the ovaries, natural pregnancy becomes difficult or impossible. Fortunately, medical science has developed fertility drugs that contain hormones like follicle stimulating hormone and luteinizing hormone. These can be administered to stimulate ovulation, the release of eggs from the ovaries. These treatments help many women become pregnant who previously couldn't conceive. However, the success is not guaranteed for everyone. Some women may need multiple attempts before achieving pregnancy, which can be expensive and emotionally challenging. Another potential issue is that hormone treatments can sometimes stimulate too many eggs, resulting in multiple pregnancies, such as twins or triplets. For couples who still struggle to conceive using hormone treatments alone, in vitro fertilization, IVF, provides another option. IVF involves collecting eggs from a woman's ovaries and fertilizing them in a laboratory using sperm from her partner or a donor. Before the egg collection procedure, the woman is typically given FSH and LH to stimulate several eggs to mature. This increases the chances of obtaining viable eggs for fertilization. The collected eggs are then combined with sperm in a laboratory dish. If fertilization occurs successfully, the resulting embryos are grown in a special incubator. For couples where the man has a very low sperm count, a technique called intracytoplasmic sperm injection may be used. This involves directly injecting a single sperm into an egg. Once the embryos have developed into small balls of cells, one or more are transferred to the woman's uterus. Any remaining healthy embryos can be frozen for future use if needed. Fertility treatment offers hope to many couples who couldn't otherwise have biological children. For those who have longed for a family, this technology can fulfill a deeply held wish. However, the process comes with challenges. The success rate of IVF is relatively low. This means that many couples face disappointment. The process can also be emotionally stressful, with cycles of hope and potential disappointment. Many find the uncertainty difficult to manage. It's also physically demanding for women who may experience side effects from the hormone treatments, such as abdominal pain, bloating, and headaches. Multiple births are more common with fertility treatments. While many parents are delighted with twins or triplets, multiple pregnancies carry higher risks of complications for both the mother and babies. Advances in laboratory techniques have improved IVF outcomes over the years. Specialized microtools help embryologists handle eggs and sperm under the microscope with greater precision. Time-lapse imaging technology now allows continuous monitoring of embryo development without removing them from the incubator. This helps identify the embryos most likely to result in successful pregnancies. Some people have ethical concerns about certain aspects of IVF treatment. The process often results in unused embryos that may eventually be destroyed, which some find problematic. Genetic testing of embryos before implantation is now possible, but raises questions about selecting embryos based on their genetic characteristics. Despite these concerns, IVF has helped millions of people worldwide become parents who otherwise might not have had the opportunity. Understanding how hormones regulate fertility has been crucial in developing these treatments. The careful application of this knowledge has transformed the lives of many families. In our next episode, we'll explore the complex topic of negative feedback systems and how they help regulate various hormones throughout the body. Let's take a look at what we'll be covering in this video. First, we'll examine adrenaline and its role in preparing the body for fight or flight. Next, we'll look at thyroxine and how it regulates metabolism. Finally, we'll explore how negative feedback systems control hormone levels in the body. Adrenaline is a hormone released by the adrenal glands, which sit just above the kidneys. It's sometimes called the emergency hormone because it prepares your body to respond to stressful or scary situations. When you're faced with danger or stress, your brain detects fear or stress and sends nerve impulses to your adrenal glands, which respond by secreting adrenaline. Adrenaline prepares the body for fight or flight by triggering a series of rapid changes. It increases blood pressure and heart rate, ensuring more blood flows to the muscles and brain. It also increases your breathing rate, which brings more oxygen into the body. Adrenaline raises blood glucose levels to provide extra energy and increases the rate of respiration in cells. Together, these changes prepare you to either face the danger or run away from it, the classic fight or flight response. Now let's turn to thyroxine, which plays a very different role in the body. Thyroxine is released by the thyroid gland located in the neck. Unlike adrenaline, which prepares us for emergencies, thyroxine regulates our day-to-day metabolism, the chemical processes that occur within our cells. Thyroxine helps control the basal metabolic rate, the speed at which chemical reactions happen in the body when at rest. It's important for regulating protein synthesis for growth and development. The hormone also supports many essential processes like stimulating protein synthesis for growth and development. Without adequate thyroxine, normal development cannot occur. Both adrenaline and thyroxine are controlled by negative feedback systems. This is a key mechanism that maintains hormone levels within narrow limits. In a negative feedback system, when levels of a hormone rise too high or fall too low, it triggers responses that bring the level back to normal. Let's look at how this works for thyroxine. The release of thyroxine is controlled by thyroid stimulating hormone TSH, which is released by the pituitary gland. When the level of thyroxine in the blood is too low, the pituitary gland secretes more TSH. This stimulates the thyroid gland to release more thyroxine, bringing levels back to normal. Conversely, when thyroxine levels are too high, the secretion of TSH from the pituitary gland is reduced. This decreases the amount of thyroxine released from the thyroid, bringing levels back down to normal. This is a classic example of negative feedback. The system responds in the opposite direction to the change, helping maintain balance. Negative feedback is crucial for maintaining homeostasis, keeping conditions in the body relatively constant. Without it, hormone levels could fluctuate wildly, causing serious disruption to bodily functions. Understanding how hormones like adrenaline and thyroxine work and how their levels are regulated helps us appreciate the sophisticated control systems that keep our bodies functioning properly. In our next episode, we'll be exploring plant hormones, discovering how they regulate plant behavior. To start off, here's a quick overview of what we'll go through in this video. First, we'll examine auxin, a key hormone that controls plant growth. Next, we'll look at how auxin helps plants respond to light and gravity. Then we'll explore gibberellins and their role in plant development. Finally, we'll discover how ephine controls fruit ripening and how these hormones are used commercially. Plants don't just grow randomly. Their development is carefully controlled to ensure they grow in the right direction and respond appropriately to environmental conditions. Plant hormones are chemical substances produced in small amounts that influence plant growth and development. They're transported around the plant to coordinate responses to the environment. Auxin is one of the most important plant hormones. It controls growth near the tips of shoots and roots, helping to determine the plant's overall shape. Auxin responds to environmental cues like light and gravity. When a plant detects these stimuli, it adjusts auxin distribution, which changes how different parts of the plant grow. Auxin is produced in the tips of plants and moves backwards to stimulate cell elongation. This process occurs in the cells just behind the tip. If the tip of a shoot is removed, no auxin is available, and the plant may stop growing. This is why pruning can control a plant's size and shape. One interesting feature of auxin is that it affects different plant parts in different ways. Extra auxin promotes growth in the shoot but inhibits growth in the root. This produces a coordinated growth pattern that helps the plant thrive. Let's look at how auxin helps plants respond to light, a process called phototropism. Plants need light for photosynthesis, so it's advantageous for them to grow toward light sources. When a shoot tip is exposed to light, more auxin accumulates in the shade than the side that's in the light. This uneven distribution causes cells on the shaded side to grow longer. As a result, the shoot bends toward the light. This bending allows plants to maximize their exposure to sunlight, which is essential for photosynthesis. Plants also respond to gravity, a process called gravitropism. This helps roots grow downward into the soil and shoots grow upward. When a shoot is growing sideways, gravity produces an unequal distribution of auxin in the tip, with more auxin on the lower side. This causes the lower side to grow faster, bending the shoot upwards. In roots, the opposite happens. A root growing sideways will also have more auxin on its lower side. However, in roots, extra auxin inhibits growth. This means the cells on top elongate faster and the root bends downwards. These responses ensure that shoots grow up toward light, while roots grow down into the soil where they can absorb water and minerals. Scientists have found many practical applications for plant hormones. They can be extracted from plants, or artificial copies can be made. These hormones are then used in agriculture and horticulture. Auxins have several commercial uses. One important application is in weed control. Most weeds in fields or lawns are broad-leaved plants, unlike crops and grasses which have narrow leaves. Selective weed killers have been developed using auxins, which only affect the broad-leaved plants. They disrupt the normal growth patterns of these weeds, causing them to die while leaving grasses and crops unharmed. Auxins are also used as rooting powders. A cutting is a part of a plant that has been cut off, like the end of a branch with a few leaves on it. Normally, these cuttings would not grow roots. However, if rooting powder containing auxins is applied, cuttings can produce roots rapidly and start growing as new plants. This enables growers to create many identical copies of a valuable plant much more quickly than growing from seeds. Another application is in tissue culture, where plants are grown from small clusters of cells. Hormones, including auxins, are added to the growth medium along with nutrients to stimulate the cells to divide and form both roots and shoots. Gibberellin is another important plant growth hormone. It stimulates seed germination, stem growth, and flowering. One major use of gibberellins is controlling seed dormancy. Many seeds won't germinate until they've been through certain conditions, such as a period of cold or dryness. This is called dormancy and helps ensure seeds germinate at the right time of year. Seeds can be treated with gibberellins to break dormancy and make them germinate at times of year when they wouldn't normally grow. This gives farmers more control over when crops develop. Gibberellins also help with flowering. Some plants require specific conditions to flower, such as longer days or low temperatures. When treated with gibberellins, they can flower without these environmental triggers, which is useful for greenhouse growers who want to produce flowers all year round. Another commercial application is increasing fruit size. Seedless varieties of fruit often don't grow as large as seeded fruit. By applying gibberellins to these fruits, they grow larger to match the size of normal types, making them more appealing to consumers. Ethene is a gas produced by aging parts of plants. It plays a crucial role in fruit ripening by controlling cell division and stimulating enzymes that cause fruit to ripen. Commercially, ethene is used to speed up the ripening of fruits. Fruits are often harvested while they're still green, so they don't get damaged during transport. The gas is then added to ripening rooms at the destination to trigger the ripening process just before sale. This means that fruit can be picked, transported, and arrive at supermarkets in perfect condition. Without this technique, much more fruit would be damaged during shipping. Ripening can also be delayed by storing fruit in conditions that block ethene's effect or reduce the amount of ethene the fruit can produce. This extends the shelf life of many fruits. Understanding plant hormones has revolutionized agriculture and horticulture. By controlling these natural chemical messengers, we can influence how plants grow, when they flower, and how quickly fruits ripen. This knowledge has helped increase crop yields, reduce waste, and develop new methods of plant propagation. As research continues, we'll likely discover even more ways to work with plant hormones to improve food production and create more resilient crops.